Hello, and thank you for joining me for this webinar on propagating the salt marsh grass Spartina alterniflora, commonly known as the smooth cordgrass. Why should your nursery produce Spartina? Spartina is the dominant salt marsh plant found here in coastal Georgia. And our salt marshes are one of our greatest natural resources in the state. Not only does the marsh provide numerous ecosystem services, including flood control and flood protection from storm events, water filtration, and carbon sequestration, they also contribute to the nursery habitat for numerous fish and seafood species, some which are commercially important to our fisheries. There has been a lot of degradation of marshes over the past decades, and in the past they used some type of coastal armoring to protect those shorelines and mitigate erosion, and particularly to protect property. But recently, efforts have started to focus on using these nature-based solutions to restore habitats. In the salt marsh, there is a technique called living shorelines that is often used to restore shorelines and mitigate erosion. These living shorelines involve the use of green infrastructure such as core fiber logs or even bagged oyster shells, and the planting of native biota to not only mitigate erosion and restore shorelines, but also restore ecosystem functions. Due to its vital role in salt marsh creation, maintenance, and ecosystem function, Spartina alterniflora is generally the plant of choice to plant in these living shoreline projects in the salt marsh. For Georgia restoration, there currently is no local source of Spartina to support Georgia living shorelines. And research has found that to increase success and restore ecosystem function, local plants propagated from seeds should be used. However, to our knowledge, there is currently no local source for Spartina in the state of Georgia for living shorelines, and that is where your nursery comes in. So producing Spartina from seeds is not a particularly hard process. The first thing you have to do is, of course, go and collect the seeds. Flowering of the Spartina often begins in mid to late summer in July and ends sometime in October, as you can see from the phenology graph created by the Joesting Lab. The inflorescences, or flowering stalks of Spartina, consist of small whitish flowers arranged as a spikelet on the upper 20 to 35 centimeters of the stem. Flowers are wind pollinated, and in about mid-September, fruits will start to form. These fruits will eventually dry and develop into seeds beginning from late September and lasting through late November. Seeds are approximately 1 centimeter in length and are light brown. Seeds are ready for collection once the stem of the seeds is brown and the seeds shatter or fall off from a gentle disturbance. Research in our lab has found that the greatest number of seeds per flowering stalk and the greatest seed viability is found if seeds are collected between mid-October and late November. So it is our recommendation that this is the time frame where seeds should be collected. Please be aware, though, that the collection of seeds of Spartina from the marsh does require a collection permit from the Georgia DNR. Once you have located a seed that is ready to be collected, place a gallon-sized sealable bag over that spikelet and remove the stem with scissors or a knife. We have also suggested that marshes with relatively low human disturbance and located further inland within the coastal region be selected again to maximize the number of seeds per spikelet and seed viability. Many spikelets contain empty seeds or sterile seeds, and not all inflorescences mature, that's the flowering stalks, at the same time. So the collection of seeds may require multiple collection visits to sites, and it was recommended that more seeds than spikelets than you think you will need are to be collected. collecting seeds, several things to avoid are spikelets or seed stalks with the signs of fungal infections. This includes ergot and rust, which is shown here. Rust is the orange coloration found on the leaves, and ergot is kind of that purple structure that is growing out of the seeds. It is also suggested to avoid spikelets with obvious human consumption, as these stressors may impact reproductive output. Once seeds have been collected, they are then returned to the lab for processing. Spikelets should be processed as soon as possible. If they are unable to be processed immediately, they can be temporarily stored by wrapping the spikelets in a wet paper towel, wetting that wet paper towel, and storing them back in their bags in the refrigerator. Seeds can be easily removed from the spikelet by hand, just by rubbing the stalk through your hands. These spikelets can be removed. And then seeds must be placed under a cold, wet conditions in order to break dormancy. These plants require at least 2 months of wet, cold conditions in order to become viable. To do this, place seeds in a sealable plastic baggie filled approximately halfway with fresh water, and then store those seeds at about 4 to 10 degrees Celsius for at least 2 months. For us, this ranges between storing them in a refrigerator, which refrigerators tend to be around 4 degrees Celsius, or if you have something like a growth chamber, you can set this at 10 degrees Celsius. This is around 50 degrees Fahrenheit and is more representable of the soil temperatures over winter in coastal Georgia. These seeds must be stored again for at least 2 months in order for dormancy to be broken and for seeds to germinate. Collected seeds are only viable for 1 year, and after 1 year should be discarded. For seed germination, please watch the below video.
After at least 2 months of cold stratification in those cold wet conditions, seeds can be removed from the plastic bag by dumping them into a sieve. We like to use the number 18 sieve that has a good mesh size to keep the seeds from falling through. Make sure that all seeds are removed from the plastic bag. You can do so by just rinsing that bag out into the sieve. And then thoroughly rinse seeds with fresh water, as seen here. Once seeds are rinsed, they can then be transferred into a glass culture dish or another type of medium for germination. In our lab, we use forceps, or famously called tweezers, to transfer seeds from the sieve into this glass culture dish. This glass culture dish will then be filled with fresh water, which is really important in germination process of Spartina. These seeds are considered recalcitrant, which means that they will die if they become dry. So water must be maintained in this dish for germination. Seeds in that glass culture dish can then be placed in either greenhouse conditions or under a growth cart and monitored for germination. Once germinated seeds have a seed stem that is about 1 inch in length, those seeds can then be transferred into Jiffy peat pellets, gently making sure that the roots are embedded and that your stem is facing upwards and not covered with sediment. These Jiffy peat pellets can then be maintained again in the greenhouse or on a growth cart in a sub-irrigated tray. The sub-irrigation allows those Jiffy peat pellets to remain wet and keeps the seed and the little seedling from drying out.
Once you can keep those seeds on the growth cart or under germination for several weeks until seeds get to about 6 inches, and then they are planted following these procedures.
When seedlings have reached approximately 6 inches in height, they can be transplanted into a 6-inch pot using a 2-to-1 topsoil to sand ratio potting mix. Remove the mesh from around the Jiffy peat pellet and carefully plant within the soil. making sure that you have plenty of soil and that the seedling is emerging from the top. You can maintain these seedlings in greenhouse conditions under sub-irrigated trays, as shown here. Making sure to sub-irrigate those with fresh water, maintaining them full, or you can also maintain them in an above-ground wetland. And in the above-ground wetlands, these can be stored for over winter, and they will also eventually start to produce flowers and seeds.
And with that, I would like to thank you for joining me. And if you have any questions or you want more information or you would like to collaborate, feel free to contact me. My name is Dr. Heather Joesting, and you can see my email address here below. I also want to acknowledge that the development of this protocol was supported through a Cycle 24 and 25 Coastal Incentive Grant from the Georgia Department of Natural Resources. This work also could not have been completed without the hard work of master students Justin Henson and Megan Palacios, as well as many undergraduate research students in the Joesting Lab here at Georgia Southern University. I also want to give a quick shout out to master student Kayla Corey, who was kind enough to serve as our planter and our video starter to show you some of these techniques. These are the citations that I have used for looking at the genetic variation within Spartina, both in populations and in other sources.